Good morning, everyone. Good morning. We're just going to get started very shortly. Thank you for all of you who've joined and signed on so promptly on your Saturday afternoon. We're really excited about today's session. Just going to give folks a couple minutes to get situated and signed on before we kick things off. Just going to give about another minute or so. Okay, without further ado, good morning, everyone, and welcome to our Innovative Cures for Sickle Cell Disease and the Drug Approval Process in Canada webinar. I'm joined by my lovely co-hosts and panelists who you can see on the screen, but I will introduce them all briefly. For get things started, my name is Delaney Hines. My preferred pronouns are he, him. I serve as both the Education Chair for Chicago as well as a member of our Board of Directors. Um, just so you know, I'll be moderating today's session as well. It will be recorded. So if you are members of your network or colleagues who are unable to participate in the live session, would like to view a recording, you'll find the recording of today's session posted on our website at https www.sicklecellanemia.ca for viewing at your convenience. I'm just going to advance my slides. One of our missions at SCAGO, as many of you know, is to serve as a conduit for members of our community to access evidence-driven, apologies, it's a bit outdated, evidence-driven, up-to-date information like we have prepared for you this morning. Today's session is important because we hope to guide you through not only innovative cures, but patient advocacy, lived experience, and some of the ongoing processes and initiatives that are being introduced to Canadian context to help folks living with sickle cell disease get access to these innovative cures and treatments. So in this session, we'll be hearing from the folks you see on my screen, Dr. Donna Wall, Victoria Gray, Dr. Matthew McDonald, and absolutely not, last but not least, Gabriel Badejo, just to give you a different perspective on the variety of different options that are available, as well as some of the lived experience that these folks have been able to undergo as they've been expanding their awareness of what's available. So our agenda, as you can imagine, we'll start with the land acknowledgement. Followed by that, we'll have some additional housekeeping rules, just so folks are aware of how to operate in today's session. And then following the presentations from our panelists, we'll have our guest speakers available to answer any of your questions or any of your inquiries. So with that, we acknowledge that the land we are meeting on today is a traditional territory of the many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee. Why is my mouse doing this? Apologies, folks. The Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, both Inuit, First Nations, Inuit, and Métis. We also acknowledge that Toronto is covered by Treaty 13, which is the territory traditionally owned by the Mississaugas of the Credit. I myself am joining you from Ajax, Ontario, which is the traditional territory of the Haudenosaunee, Huron-Wendat, and the Anishinaabe Mississauga, adjacent to the Mississaugas of Scugog Island First Nation, covered by the Williams Treaty. And I'm really privileged to have the opportunity to serve as your moderator today. So I also want to make note, like I mentioned before, with respect to the housekeeping, the session will be recorded. If you have any questions, please feel free to use our Q&A or chat feature. If you'd like to come off mute and speak directly with our speaker, you can also raise your hand, and either myself or one of my colleagues will go through the actual participant list and try our best to remove you from being muted and enable your audio. And as you can imagine, we have a pretty large audience today, so we will do our best to go through as many questions as possible. But if there are any questions we're unable to attend to, please feel free to email me. I can put my email in the chat. Otherwise, I'll continue as proceeding. So, without further ado, I would like to introduce our first speaker, Dr. Matthew McDonald. Dr. Matthew McDonald is the Acting Director of Pharmaceutical Policy and Health Technology Assessments as a part of the Evidence Products and Services Business Unit at CADTH, which is the Canada's Drug and Health Technology Agency. He's been involved in the launching of CADTH's non-sponsored reimbursement reviews and the Formulary Management Expert Committee. Both initiatives provide tools for payers to reimburse new indications for older drugs or revisit previous recommendations for older drugs to increase treatment options. Prior to being at CADTH, Dr. McDonald was a research fellow at the University of Ottawa, where he researched novel approaches to promote the restoration of motor function following stroke. So today he'll be providing us with an overview of the drug reimbursement process in Canada, the role of CADTH, and highlighting some ongoing initiatives to improve treatment options. Following up Dr. McDonald, we have our heavy hitter, Dr. Donna Wall. She joins us from the Hospital for Sick Children or Sick Kids and University of Toronto. In the fall of 2016, she signed on as a section head of the blood and marrow transplant / cellular therapy program. At Sick Kids, she's involved in bone marrow transplant and gene therapy trials, utilizing CRISPR and Cas9 editing of hematopoietic stem cells to treat thalassemia and sickle cell disease. So today she'll be giving us an overview of what's involved with current gene therapy strategies and early outcomes, and she'll briefly describe the approval process for new treatment strategies. Following us next, we have a great session and a great delivery from Victoria Gray. She was diagnosed with sickle cell anemia at the age of 3 months. She became the first patient in the United States to enter a clinical trial using a CRISPR gene editing drug to treat her sickle cell disease. Almost 5 years later, she and dozens of others from the trial are now doing amazingly well. She's been able to experience motherhood, working full time, and become an advocate, and she's here joining us today to share her experience. She was recently invited to be the keynote speaker at a genetics conference celebrating the success of the treatment and exploring the remaining hurdles of gene editing. And like I mentioned before, last but absolutely not least, we have my guy, Gabriel Badejo. He's a Toronto-based Afro-Canadian producer, director, and sickle cell anemia activist. After graduating from the Toronto Film School, he landed a production assistant role on The Next Star, and his career took off. He's worked with major brands such as Google, Converse, Land Rover, and the award-nominated CBC Gem series, The 410. As a Hot Docs Doc Accelerator Fellow, he's currently working on his first featured documentary, which I'm sure we'll be able to broadcast later. Gabriel's lived experience is currently part of a team working to improve experiences in emergency departments province-wide as a member of the Ontario Quality Standards Committee. And today he'll be telling you his story and his experience with bone marrow transplants. Okay, I've done enough talking. Want to kick things over to our main guests and our main folks we've come to hear from. So over to you, Dr. McDonald.
Sure, thank you very much. Let me just quickly share my screen here.
Perfect.
Flip this over so that it looks bigger.
Yeah, we're seeing the what do I call it? We have to flip it back. It's currently in your present. There it is.
Yeah, there we go.
There we go.
Perfect. So everyone can see a little better.
Yeah, there we go.
Excellent. Yeah, thank you very much for the opportunity to come speak to everyone today. So really the goal of my talk is to provide an overview of the drug reimbursement review process in Canada. So the goal of my talk really first is just kind of set the stage, is to provide an overview of CADTH, what is our role in drug reimbursement in Canada, provide an overview of the drug approval process in Canada, as well as the specifically reimbursement review process. And lastly, I'm going to outline some ongoing initiatives to improve treatment options and some opportunities for engagement from our stakeholders. So just starting with a disclosure statement. So CADIF is funded by contributions from our Canadian federal, provincial, territorial ministries of health, with the exception of Quebec. And we also receive application fees from the pharmaceutical industry for a variety of our products. So just to level set us today, so what is CADIF? So CADIF performs health technology assessments for publicly funded healthcare providers. We're an independent, not-for-profit organization. So what is a health technology assessment, or HTA? It's really a multidisciplinary process that uses explicit methods to determine the value of a health technology at different points in its life cycle. So by life cycle, what I mean is this could be a drug that is new and not yet available, or potentially it could be a drug that's in the post-market space that's been around for a period of time, and potentially new evidence has emerged to inform its place in therapy. So what does CADIF do? So as I mentioned, we provide health technology assessments to allow decision makers to make evidence-based policy decisions. We complete work in a variety of spaces, specifically pharmaceuticals, medical devices, clinical interventions, as well as health systems. Types of questions that are generally addressed through our work: how effective is a new drug compared to other treatments? Is it better than what we already have? How accurate is a new diagnostic test? Should a new product be reimbursed by public payers? Which patients would benefit most from a particular treatment? What's the appropriate price for a product? As well as outlining any implementation barriers for new health technologies. Sorry, I keep looking to my left because I have the Zoom camera over here, so it feels more like I'm talking to someone. Okay, so in terms of the Canadian drug landscape in Canada, so it is rather complex, but it can kind of be broken down into these 4 buckets. So starting first with Health Canada, our federal regulator. So an industry sponsor would decide they would like to market a drug in Canada, and they would submit an application to Health Canada for approval. And Health Canada, as the regulator, really looks at, does the drug work? Outline any risks or potential side effects with the treatment, and really look at, are the benefits greater than the risks? Or can the risks be properly mitigated? As well as Health Canada looks at the manufacturing process of the drug to make sure that it's also of high quality. And at the completion of their review, if the benefits kind of outweigh the risks and the risk can be mitigated, the drug's ultimately issued a notice of compliance, so an NOC, and it's given a drug identification number, which permits the sponsor to market the drug in Canada. Next, concurrently with the Health Canada submission, or shortly after, industry sponsor would also submit to a health technology assessment body like CADIF, or in Quebec, INES. And what CADIF really looks at is, how does the drug compare with existing treatment options? Is it good value? If there's no existing treatment options already exist, how does it compare with the current standard of care? Importantly, we try to identify, who does it work for? So what patients would benefit most from the treatment? What's its value? Is it cost effective? As well as working with our stakeholders to try and identify any implementation issues around the new drug. So for example, is there any health human resource issues? Is there issues around location of care associated with the new treatment? Also, another important player in this space is the Patented Medicines Prices Review Board. So PMPRB, so it's a long acronym there. So it's a federal agency that really regulates and reports on the prices of patented medicines in Canada. And really the goal is to ensure that prices of patented medicines sold in Canada are not excessive. And then lastly, what we have is the public drug programs as well as the PCPA. So the PCPA is the Pan-Canadian Pharmaceutical Alliance. And really, the final funding decision is left with the public drug programs, and they work in consultation with the Pan-Canadian Pharmaceutical Alliance to negotiate a price. And what they really try and determine is the drug needed and is it affordable? So in Canada, we have a number of different public drug programs that are federal, provincial, territorial, also some blood product formularies. There's also numerous hospital assessment bodies as well as funding bodies. So specifically CADA, so when we receive a submission, we perform what's called as a reimbursement review. So a reimbursement review is a pan-Canadian process that provides reimbursement recommendations and advice to publicly funded drug programs. Really, the goal of this process is to reduce duplication across jurisdictions, maximize the use of our limited resources, and enhance consistency of drug reviews and reimbursement decisions. So what does a drug reimbursement review process look like? So it starts with a submission from a drug manufacturer, which would outline the drug under consideration for public reimbursement and providing any clinical data or information to inform on its efficacy. So once CADTH would receive a submission from a sponsor, we perform a review, so a reimbursement review on this submission in consultation with our stakeholders. So that's both the public drug programs through some of our advisory committees, as well as patient groups, clinician groups, and clinical experts in the given area. What does go into the CADTH review and what's considered? There's a number of components. There's the clinical review that looks at randomized control trials, any indirect comparisons for the treatment under review. Also some other evidence, such as real-world evidence that could look to fill evidence gaps that exist through the RCTs, randomized control trials. Also the economic review. So there's a economic review is submitted by the sponsor. We look to validate the data and any assumptions made in their submission and really provide information on the affordability of the drug under review. In more complex reviews, we also incorporate an ethics review to try and identify any ethical considerations around the drug under review. And lastly, and most importantly, is we try and engage stakeholders early and throughout the reimbursement review process. So we do that through a number of ways. First and foremost, any drug that is submitted to CADTH, we have an open call for stakeholder feedback where patient groups, clinician groups, public drug programs can provide feedback on the drug under review. They complete standardized templates. They often conduct surveys or interviews to help inform the submission to CADTH. All the input that we receive conflict of interest declarations, which are posted for transparency. We work with the drug programs to try and identify early any implementation issues that should be considered by the review. We also engage with clinical specialists. So each of our reimbursement reviews has at least one expert in the area under review. And the expert really has a number of important tasks in the review. They help us provide appraisal and interpretation of the clinical data. They really help us try and characterize the unmet therapeutic need, identifying any gaps in evidence or opportunities for data collection, any implementation challenges, really provide their perspective on the clinical management of patients that would benefit most from the drug under review. And they also participate in the expert committee deliberation during a Q&A session. We also engage with patient groups through submissions, as I mentioned earlier, sometimes providing surveys. Really what we're looking for from the patient input in our reimbursement reviews is really trying to better understand the impact of the illness on the person's life and really try and identify the appropriate treatment goals. What means the most to patients? The input helps us to explore if the evidence within our review addresses the needs of patients with lived experience. Also helps us contribute to the interpretation of clinical trial evidence, the identification of gaps, and also to help us identify use, equity, as well as ethical considerations that should be considered. So really the goal is to try and capture the patient voice. We really want to hear from individuals and caregivers with lived experience so that we can communicate this in the CADTH review as well as in the final deliberation and recommendation. So once we perform the CADTH review, this goes to one of our many expert committees at CADTH. And as I mentioned, we have at least one clinical expert that would attend the deliberation and is given a question and answer period where the expert committee can ask the clinician specific questions. We also have a patient member who's part of the core committee, and they take part in the entire review and deliberation and voting on the final recommendation. So the output of the expert committee deliberation is ultimately a reimbursement recommendation to our public drug programs. And the recommendations, as I mentioned, are provided by a number of our expert committees at CADTH. So we have committees around specifically oncology drugs, non-oncology drugs, plasma protein products, as well as reviews more in the post-market drug space, so which is our formulary management expert committee that I'll touch a little bit briefly on later. So really the recommendations provided guide funding decisions, but it's important to recognize that they're non-binding. The final funding decisions do remain the responsibility of each individual public drug program. The recommendation that comes from our committees can be categorized into three ways. So reimburse with conditions. So an example of a condition would be identifying which patients would benefit most from the treatment under review, or potentially a do not reimburse recommendation. So with the last little bit of time that I have in my talk today, I just want to discuss some kind of ongoing initiatives that we have going at CADTH and some opportunities for stakeholder engagement in this space. So I thought I'd first start with time-limited recommendations. So this is a new initiative that we've launched at CADTH. So as I mentioned earlier in my talk around Health Canada and receiving a notice of compliance, in some situations, Health Canada would grant a notice of compliance with conditions. And this is authorization to market a drug with the condition that an industry sponsor undertakes additional studies to verify the clinical benefit of the drug. So in this space, to help ensure timely access to promising new therapies and ensure that our recommendations accurately reflect the currently available evidence, when drugs receive a conditional regulatory approval, we now have the avenue of a time-limited recommendation, which would be issued, which is really an issue of a reimbursement recommendation that's contingent on the future assessment of evidence to try and address any uncertainty with the comparative clinical benefit or cost effectiveness. So at this point in time, we're trying the time-limited recommendations process in a small subset of files and engaging with our stakeholders through the process to try and establish and refine any implementation considerations around a time-limited recommendation. Next, I'm going to talk a little bit about the Formulary Management Expert Committee. So CADTH is committed to evaluating technologies across their life cycle. So not just for new innovative drugs, but also going back as new evidence emerges and evaluating their place in therapy. So this past year, we launched a new pilot project intended to provide expert recommendations for payer-initiated drug reviews in the post-market space. And in this committee also, we're using a real test and learn approach to try and pilot some new innovative concepts to how we conduct our reviews in terms of how we deliberate on our reimbursement reviews and really what the final output of a review might look like. So this figure here is just showing 5 kind of concepts that we've worked and piloted through our new expert committee. So one of the things is the types of reviews that would get a reimbursement recommendation. So the Formulary Management Expert Committee also looks at drugs that are at or beyond their exclusivity status. So perhaps the drug's no longer branded and we have generic entrants of a drug. And new evidence is available to support its use in a previously unfunded indication. This committee could then review those kind of situations. We also are looking at reviews that reevaluate the place in therapy of drugs that are already publicly reimbursed. So there's always a lot of research going on. So it's important that we have an opportunity and an avenue to go and reevaluate drugs that are already marketed in Canada to make sure that they're being used in the best manner. We're also looking at our deliberative framework and process. So how can we deliberate in a more transparent manner? And how can we capture the patient voice more in our deliberations? So we're trialing some different approaches with how we deliberate, also really trying to capture dissenting opinion as well, dissenting opinion from the members of our expert committees, and also trying to identify specifically how the patient voice and patient input is incorporated into the CADTH review, but also how does it contribute to the final reimbursement recommendation. We're also trialing a lot of new concepts around stakeholder input. So looking for ways on how we can incorporate stakeholders more into the process. So for example, we have our clinical experts now take part fully in the deliberation. As I mentioned previously, normally we have a question and answer period for the experts. We're exploring having the experts around the entire time so that they can really provide their perspective. And we're also incorporating the patient voice a little more. So at our expert committees meeting, we're given the opportunity for a patient with lived experience to come to the committee in deliberation and actually present their story to the committee so that we also get that kind of personal perspective, that personal story incorporated into the deliberation as well. We're also looking at what the review outputs look like, so kind of their look and feel. How do we conduct our reviews, but ultimately, what does the final output of a reimbursement recommendation look like? And trying to, again, really focusing on being more transparent, recording votes, providing summaries of dissenting opinion, but most importantly, really trying to improve the readability of our reports and trying to keep them very succinct and digestible from all different types of audiences. And lastly, I just wanted to touch on the transparency piece, which really overlaps with all of these concepts. And as we kind of go forward with the Formulary Management Expert Committee, we're looking at how can we better improve our transparency in terms of the CADTH review, in terms of how our expert committees deliberate, and in terms of how the committee ultimately got to their final recommendation. So I'm just going to end my talk today on just highlighting the CADTH 2024 watch list. So this is an annual Horizon Scan report that looks at emerging health technology trends that are likely to disrupt the delivery of healthcare in Canada over the next 5 years. to really try and give decision makers an early signal to help guide health system planning. So the theme of this year's watch list is care for children and youth with medical complexity, which is obviously a diverse group with a range of complex needs that can involve a reliance on technology and other support systems. So we will have a webinar this next Tuesday, March 5th. I provided the link here, but it's at March 5th from 1 to 2. And you can also find this on our CADIT website as well for those that are interested. So that's all for me for today, and thank you all for listening.
Much appreciated, Dr. McDonald. What I'll do, folks, is I will grab that link from the website, from CADIT's website, and post it in the chat so folks can get access to that webinar, followed by also letting them know about the call for nominations for any of the patient advisory committees folks may be interested or able to participate in. Really appreciate the comprehensive overview. I always take away a lot from all the different ways that CADIT is trying their best to once again bring these interventions to people at a cost, with the cost consciously. But thank you for that. I'm sure there'll be some questions later on today. So without further ado, I wanted to transition now to the presentation that Dr. Donna Wall has developed for us. As you all know, she's a section head for blood and marrow transplant and cellular therapy at the Hospital for Sick Children, as well as a clinical researcher, and she'll be taking us through gene therapy for treatment of sickle cell anemia. So over to you, Dr. Wall.
Well, thank you very much. That was a tremendous talk. I've had the privilege of working with CADIT over the years, and I tell you, CADIT is a true Canadian gem, and the quality of the reviews are absolutely spectacular. And I'd strongly encourage people on this call to not be afraid, but to get actively involved in bringing the voice of the community to CADIT. I think what we really want to do is hear from the next speaker. So I'm going to set her up and give you some background information about what's involved with gene therapy. I've been working on this for the last 5 years heavily, and have been truly humbled with the complexity involved and the true benefit that can come from these types of therapies. So just quickly, I just have to let you know I'm running trials for with the CRISPR Vertex therapy at sick kids, and I do provide advice for Vertex and Aditas. I do not get any money for this work. Okay, so I just got to get everybody up to speed quickly. So this is my life in a nutshell. We make our blood inside our bones, and the whole blood making system is driven from stem cells or mother cells that live in our bones and give us lifelong blood making. That blood making makes our red blood cells, where the sickle cell problem is, makes white blood cells that and platelets keep us from bleeding, and the white blood cells give us our immunity. When we do a bone marrow transplant, we bring in somebody else's young blood making system, their mother cells, and basically replace the whole system. In doing so, we have to deal with arguments between the donor cells and the patients with the new immune system. We do replace out, all parts of the blood making system. With gene therapy, the idea here is we target these young blood making cells, not from a donor, but from the patient's own young blood making cells. And we engineer them so that they're no longer going to make cells that sickle as frequently. Okay. So when we do transplant for sickle cell anemia, it's and gene therapy, it is very it's complicated, and for something that should be so straightforward and so simple. We know exactly where the amino acid change is. We know exactly what nucleotide gets changed. But everybody with sickle cell has a very different journey, very different story. And so this is what's going to make it difficult over the next few years and trying to figure out who should be treated and when they should be treated. With treatments, treatments such as transplant, we do carry risk with the transplant itself, and we do carry risk with anything that a patient has had coming into transplant. If they have kidney trouble or if they have lung trouble, there's a good chance we're not going to get that fixed. Okay, so I'm just trying to paint our picture of what we're dealing with. Many patients do not have a transplant option. And so this is where the gene therapy becomes very important. Okay, the gene therapy that we're going to predominantly talk about today is one where we take advantage of a kind of a unusual event that happens as we are made and as we are born. When we are in the womb, the kind of blood we're making is baby blood or fetal blood. And the reason is, there's lots of reasons that I have to be careful. I like to talk. And so to give me 15 minutes is very dangerous here. But there is a switch that happens right around the time of birth where you switch from making baby blood to making adult blood. And with that switch, the key to this is that the sickle is only in the grown-up blood, not in the baby blood. So that's why often there's a little honeymoon period when an infant is born with sickle cell disease. They're picked up at about 3 months of age, right around that time point where there's a switch off of making baby blood and the adult blood comes on. We do know from lots of experience that if by chance you have sickle cell disease and you happen to also make more fetal hemoglobin as an adult, the more fetal hemoglobin you make, the less problems you have with your sickle cell disease. So from that, the people designing this treatment said, well, what if we just make you so that you make baby blood for your life? And that is actually something we actually study. Some rare families that have something called hereditary persistence of fetal hemoglobin. Basically, they're just born making fetal hemoglobin. And sure enough, when we take a look at how they do, if you happen to be one of these people, you'll have very mild or no symptoms from the sickle cell disease. So we have this clue from our clinical medicine. Oops. And so what the strategy with the gene therapy is to look at the switching mechanism. And this switching mechanism is incredibly complicated. But the target that is used in the gene therapy is for one of the essential elements that is involved with making that switch from baby hemoglobin to adult hemoglobin. And basically the idea here is if we just break up this machinery, we won't make the switch. And so the gene therapy that we're going to talk about later here is where we target specifically just right in a very targeted way where that switching mechanism happens. And the new tool that we have, Nobel Prize went for it just a few years ago, was the development of an ability to come in and in a really, really targeted way, specifically find an area in the gene. And so in this case, that's that CRISPR-Cas, we target the BCL11A control point, and we stick to it. And then we have attached in an enzyme. An enzyme is something that breaks things down. So everybody who talks about caspases goes like this because it's like a scissor cut. So we make a scissor cut in here, and we basically just quiet down that gene. And with that, you don't make the switch, okay? To do that, we collect the patient's own young mother cells or stem cells. We take them to the laboratory in a dish. We run an electric current through it. It's called electroporation. and that makes the cell membranes leaky, and that lets this complex come in, and it's an enzyme reaction. It happens in a couple of hours, and those cells click, click, snip, snip, and they won't make fetal hemoglobin again. So key to this is that we actually correct the youngest of the youngest mother cells, the ones that give lifelong blood making. So what does that journey look like? First, I'm going to take you to the bottom of the slide first. We have very heavy pre-treatment phase. Okay, so usually I talk to my families minimum 5 consultation sessions, explaining, talking, thinking, is this the right thing, right process? Is it the right time? What's involved? Because this is a true partnership. This is, this is a new treatment. We have to talk about fertility preservation, okay, because right now the treatment involves very strong drug called busulfan that will lessen ability to have children later on, but we have ways of preserving fertility. We also go through a period of giving more intensive transfusions. And I use this period, I call it prehabilitation. We take care of everything that we can take care of, get infections under control, get pain management, spit spot, and get ready to go. The first step, so that's screening and prehabilitation. We then collect the young blood-making cells through a process called pheresis. This can be pretty tricky in sickle cell disease, and it can be pretty laborious. Often we need multiple collection attempts to do this, but basically each collection attempt is 3 days of collection on a machine that pulls blood from one vein, spins it very quickly, gives everything back, and we just select out the young blood-making cells. So we collect them, and then send them for manufacturing. That manufacturing, first thing that happens, and this is in a specialized, basically drug manufacturing facility, we pull out the young blood-making cells, we do that CRISPR treatment, we freeze them, we then go through intensive testing, screening, making sure that it's clean, the cells are working, they've been corrected. So this is really, really high tech. Now we're ready for the treatment. If we just gave those cells to the patient, just put them into the vein, they would know where to go, but if there's no space for them to set up shop and grow, we have to give them a survival advantage. And to do that, unfortunately, right now in 2024, we have a drug called busulfan. So that's a chemotherapy drug. It's a very strong drug. You get 4 days of that. I think my next speakers will give more information about that maybe. And then once that's all out of the system, we just give the infusion of the corrected cells. It looks like water. It's a little nerve-wracking to watch it, a couple tablespoons of water, and that does it all. That says it all. We then have about a month-long hospital stay as we wait for those seeds, those young mother cells that we've given that are starting to set up shop inside the bone, expand up enough to make blood. And we're counting on these guys to make blood lifelong. As part of the study, there's close follow-up and monitoring. These are the results presented at the last large hematology meetings. And I'm going to just walk you through the slide here. Green is good, okay? And so this is looking at the length of time without pain crises. All patients on this first study had absolutely really terrible sickle cell journeys, okay? I'm going to leave that part. I usually chatter a little bit about that, but I'm going to leave it to the expert who's following me. But pain crises that have you come to the hospital that need intervention between 2 and 8 per year, and over every year for the last 2 years were what was needed to get onto this study. Okay. And so these are patients who have had a really, really rough time. The the red here is how long it takes to go without till the last red blood cell transfusion and that goes to count recovery. The I don't know what it's going to look like to you, kind of purplish bar is the time for the transfused blood to get out of your system. And then all the green part is where you're no longer, you're just going on the blood that's being made by the cells that we gave to the patients. Okay. This is a complicated journey and it's a tough journey. Okay. So there's you just got to be careful here. So, it's not a quick fix. It gets complicated. But basically we had lost one person because of COVID. But otherwise everybody else got through and there's some really nice stories in development. This is the data, guys. Okay. These are the patients treated on this study. So it's not thousands of years and it's not hundreds of years of experience. This is incredibly exciting data. It's incredibly new. Okay. Now what we see when we look at the blood is that first of all, patients' hemoglobins are spectacular, up in the 12 grams, 120 grams per liter in Canadian talk. Yeah, 120. So I've got a young man. Yeah, so I'm not even. I can tell stories, and I'm not going to. But what we do find is that the fetal hemoglobin is high. It's above 40%, and so that's pretty much very close to what a person with sickle trait has. Okay, for their amount of sickle is similar to that of a trait patient. But importantly, there's a little bit of F in all the cells. And so what that means is there's less tendency to want to sickle or to be able to sickle. And what I'm interested in is, well, it was very nice that we could do it, but does it stick? Okay, will that correction that we made, were we able to get it into the youngest of the youngest blood-making cells? And this is the thalassemia data, and the sickle cell data is looking the same. The answer is yes. Okay. What we have, the correction we have at 6 months is sticking. It's there at 12 months. It's there at 24 months. We now have some patients that are further out. So we're really, really getting excited here that we were able to get a durable correction. There are other gene therapy strategies coming. There's another CRISPR strategy run by a company called Editas that has trials opening in Canada that is using the same fetal hemoglobin trick. It's got a different target, but all other bits and bobs of what I described happen. So it's just where that specific target is. And then the other type of gene therapy you'll have heard about, beta cell, all their names are changing on me. And so it's hurting. But basically, this is a situation where instead of using the CRISPR, this time we use a virus delivery system. It's called a lentivirus that actually puts DNA in and directly modifies the cells. And so with that, we change the hemoglobin being made. So we're not trying to get more fetal hemoglobin made. We're changing the hemoglobin so that it no longer will sickle. And what that does is we are able to track the little bit of DNA that we put in. We can track the number of cells, the correction that happens over time. Again, same kind of story here with, you got to collect the cells, you got to send them for manufacturing, you got to get them, you have the same busulfan, you have the same transplant admission. So there's still a lot of study, a lot of I'm hoping for improvements yet in the tools that we have. But what we can say now is we have the proof of principle studies that are showing that this really can work. My job is to worry about, is this among others, like, is this safe in the long term? Okay. And I would not be able to face myself if I didn't mention this at this talk. There has been a signal that there may be an increased risk for leukemia. That has been seen in the gene therapies with the lentivirus products. It has there is no signal, and the gene guys have looked very, very carefully at these patients, and at anyone who's had lentivirus treatment. There's no sign that that virus is putting something into an area where the leukemia hotspots are. But in the study of this, there was people went back to the literature and went back to understanding sickle cell anemia. And with sickle cell anemia, with no treatment at all, there's a little increased risk of developing leukemia. And part of that is because, gosh, when you have sickle cell disease, you're making blood hard every day, and you've got a lot of inflammation, and you're driving that blood making system, and that sets you up for a mistake. My concern is that when we do our process for how we currently do gene therapy, whether we're going to take the patient's own blood making cells that have already been stressed for several years and put them through a manufacturing and then make them expand up to refill the whole blood making system, whether we're going to be at risk down the road. At this point, we just have to say we don't know. We're too early, okay? But I just wanted, because there's so much excitement about this treatment, I just wanted to say, hey guys, sometimes we just gotta, take a beat here and just be cautious. Now, there's no question in my mind that there are patients who would really benefit from this treatment. Okay, they have they're very sick from their sickle cell disease, very high risk for getting into serious trouble or living a life of misery, no question. But there are a ton of patients with sickle cell anemia who are doing really well with hydroxyurea and the new management tools out there. So honestly, there's a lot of discussion going on that, engaging, patients and families in everything for sure. But also, it's the hematologists, the emergency room docs, the transplant docs, the gene therapy docs, the whole care team in trying to help with this decision making. I'm very cautious, and we have many counseling sessions with families before we proceed either with bone marrow transplant or gene therapy. And this is still very much a space in action. I've already done that shout out. I want you to get to the real good speakers that follow me. But this is the work we've done at Sick Kids and UHN. It's a big team. And I have to tell you, I've really enjoyed working with SCAGO and my patients and families, and have learned so very, very much. And so I'm going to stop sharing.
You're the best, Dr. Wall. I mean, we couldn't be able to pull off sessions like this without your support and unwavering delivery of knowledge and intellect. And honestly, hearing from you every time, just shows how much transformative work you're doing and blazing up new trails to make the living conditions for folks who are living with sickle cell disease that much better. So on that same vein of trailblazing, we have in our midst a fantastic trailblazer in Victoria Gray, all the way from Forest, Mississippi. without, I don't want to go and say anything more because I want to pass the virtual microphone over to you to help us understand your journey and impart some wisdom. It's over to you.
Hello, everyone. Well, I want to give you my lived experience first with sickle cell disease and what led me to gene therapy. I received my diagnosis, as said earlier, at 3 months old. My mother, she was giving me a bath, and I started to cry. She couldn't soothe me. So we went to the emergency room. She initially thought that I was having pain from my shoulder because the doctor did dislocate it during my birth. So we was expecting, she was, because I'm just crying. She was expecting a quick fix, but that day she received the diagnosis that I had sickle cell disease. And I was in my first crisis and I may not live no more than 7 years. So she was hit hard with everything that day. And being that my onset was 3 months, I was very familiar throughout my childhood with pain. My life was tethered to emergency rooms, regular doctor's appointments with my hematologist, and pain. It would come out the blue. I could have a day at school being normal, making plans to come home, play with my brother, my best friend. And before I can get my foot in the door, one arm would start hurting, it's aching pains, and it's electrical at the same time. So you have, in a matter of minutes, it shifts and travels across my chest, and now my other arm is hurting too. Now my legs are hurting. So, and I couldn't do anything about it. I would go to my grandmother, and she knew then she had to take me to the emergency room to get relief because the crisis was just too bad. I couldn't stand. And those visits required IV fluids, blood transfusions, pain medicines, and over a week stay sometimes at the hospital. And along with that came all the restrictions. All the dreams I had as a kid got canceled due to my sickle cell. When I wanted to become a cheerleader, my doctor, no, that's too strenuous. Basketball, no, that's too strenuous. Track, no, that's too strenuous. You're going to constantly be in pain. And even I tested my limits, trust me. I would get out there and climb a tree with my brother and run around the yard, but I would easily get fatigued. And sometimes once I got up that tree, I was trapped for a little while, waiting on my brother to come rescue me because I had just run out of energy, but I didn't want sickle cell to define me. So I tested my limits despite what the doctor said. And a lot of times doctors were right, but I wanted to see for myself. I was relentless. And that was as a kid. Once I graduated high school, went to college, I initially wanted to be a doctor, a cardiologist to be exact. But going over how long it would take and all the classes and all the extra stress, I knew it was something that would be super hard for me to pursue and would take me double, if not triple the time to reach that dream. So that was another dream I had to give up, and I pursued nursing. I began trying to be a nurse. I took all my prerequisites. And then in 2010, I had one of the worst crises ever. This crisis, it took away the use of my arms. I couldn't even lift them. It took away the use of my legs. I couldn't get up to use the bathroom. I was just bound to that bed in pain and totally dependent on the staff and my family to care for me. That stay was one of the longest. I was admitted to the hospital for 3 months. The pain went on, and even once the pain stopped, I had to go through therapy and rehabilitation to learn how to support my weight on my legs again, because the pain had made my muscles very weak, and they began to shrink. So it was hard trying to support myself. That took a long time. So after that, I gave up my dream of nursing. I did become a mother. of 4 children. One is my stepdaughter, and I have 3 biological kids. And they were my next dream, just to be a great mom, a great wife to my husband. And sickle cell affected that dream too. I was constantly being pulled away. The hospital stays that was 3 times a year has turned into hospital and ER visits once a month at least, and sometimes twice, because I got an infection in my port, and after that infection ravaged my body, it went on for over a month without them finding out what it was. My body never was the same. I was constantly having crisis and weakness. And this was 2015 when I became completely homebound. And I had to get a nurse came in to my home to care for me. She had not just the basics, but she had to help me get in and out of the tub. She had to help me clean my house, get dressed, wash clothes for my kids and I, because my husband, he's in the military, so he was away. And so I didn't feel like I was even being a good wife or a good mom to my children. And I began to lose hope, because going in and out of the emergency rooms, and with sickle cell disease, there's a lot of stigma here in the US, and you start getting treated as you're drug seeking. And so the 30-minute waits that I had in the ER as a kid now become 6-hour waits in the ER as an adult, because they don't believe I'm hurting. That sent me into depression, which triggered my hopelessness. And I started giving up pretty much. But one day, my son's teacher, he called. She said, "Jamarius is acting out. I want to have a meeting with you in person." So I made my way to the school, and she told me everything he had been doing. And she took a deep breath. She said, "Victoria, I'm just going to let you know, I understand why he's acting out. I know the reason." And I asked what the reason was. She said, "Because Jamarius thinks you're going to die. So he gets in trouble so he can be at home to watch you, because he feels like one day you'll come home from school and you won't be there." That conversation crushed me, because I saw then how me not being here in my life was affecting my children already. So I had to gather the strength to keep fighting. I began doing my research on bone marrow transplant, and I saw a story about the haplo transplant, where your donor does not have to be 100% match, but my closest match would be a sibling or a parent, so my body wouldn't reject them as fast as it would reject a stranger. So I had a bad crisis in 2017, and it put me back in the hospital on stronger pain medicines. I had a ketamine pump in one arm and the Dilaudid pump in the other. Ketamine is a horse tranquilizer, pretty much. But I still felt pain. I was still attached to the bed. I couldn't move. I was hurting, and I felt like my life was just fading for me. So I had to call the head hematologist, all my nurses who was treating me at the time, into my room, and I asked them, "Do you know anything about haplo transplant or anything out there that can help me?" They all looked at one another and then looked at me and shook their head no. So I began to sink even more. But I came home and I went back to my roots, and that was my faith. I prayed to God and I asked, "Lord, give me something just to be here for my children, at least to see them through adulthood and see my husband through." And I didn't want to leave that gap between us and the kids. I didn't want to miss the weddings, the graduations. I didn't want to miss anything. I wanted to be there for all those moments. In about less than 30 days, I received a phone call from my hematologist. I said that she had some good news for me. She wanted to share it with me. on my next visit, which was in January 2018. When I got there, she said a doctor had reached out to her and asked if she had any patients that was interested in a haplo transplant. And she immediately thought of me because she know I had been searching. She told me I would have to travel to Nashville and that my brother was my closest match because we had did gene testing before. So my brother, my husband, and I, we all traveled to Nashville, and we met Dr. Haydar Frangoul. And his presence was just refreshing to me. You know, he assured me that he could do this haplo transplant for me because he had been doing bone marrow transplants for years. He's a hematologist, so he was very familiar with sickle cell, and he didn't question my pain. He didn't frown up at my story. So I felt comfortable. We began the normal testing for bone marrow transplant. My brother and I, we did the got a graft of our bone marrow, blood work, all the works. And I began going back and forth between Mississippi and Nashville doing my lung testing, heart test. And I was already on apheresis, which was when they took my blood out through a machine and replaced it with donor's blood to keep me from going into crisis as often. But that procedure would make me weak. So on one stay in Nashville where I had to just do some regular testing, I ended up going in crisis and I had to get admitted there. Dr. Frangoul, he already knew my fear of graft versus host disease from doing the haplo transplant with my brother because there's not 100% match. I would be at risk for that. So I was fearful of trading in what I knew, which was sickle cell disease, for the unknown graft versus host. So that was, it made me hesitate. So when he came to my bedside and presented CRISPR, he said, Victoria, we have a new trial that's about to begin. Have you ever heard of CRISPR? And I said, no, Dr. Frangoul. He broke it down. This is how he told me. He said, it's like, imagine having a textbook with thousands of words in it, and there's some words that's incorrect. It could go in throughout all those words and target those words that are incorrect and do a specific edit. And he gave me a video to watch about the technology. I only had 2 questions. And my first was there a risk of graft versus host? And he explained no, because these are my own cells. My body wouldn't attack and reject itself. And I asked, because I would be the first, if it didn't work for me, would I still have the option to do the transplant with my brother? And when he said, yes, you can do that transplant within a year if this has no effect on you. So it was a no-brainer for me. I gave him my yes that day, but he didn't get a solid yes. So I came home in prayer and asked God, if this is the journey, you have the gateway to my miracle. Give me peace in my heart. Don't let me go in fearful. Needless to say, I began my journey, went into the hospital for admission there with a smile on my face, outfits in play, and with joy in my heart, feeling that this was going to be for me. They harvested my cells. I got the shot in my arm to mobilize my stem cells into my bloodstream, and I began the collection days, 3 days each time, and I had 2 rounds, so 6 days total of collection for me. And after they sent them off, did the edit, and the cells was ready, I got admitted into the hospital to undergo chemo. So I did 3 days of chemotherapy with busulfan. In the days I received the chemo, I was like, oh, my dad was with me because my husband, he couldn't come in the beginning because of his job and military duties. I said, Dad, nothing is happening to me, so this may not be as bad as I thought it was. So I did the 3 days chemo, then 4 days of rest, and infusion day, July 2nd, 2019. Dr. Frangoul had 3 vials of this clear liquid that seemed to glisten. He pushed them into my port that I had in my chest real fast. It made my heart rate speed up as he was pushing because it goes directly to my heart to get pumped all over. So I was a little nervous about that. But after the 3 valves, I looked at Dr. Frankel. We did a high five, and I'm like, that's it. And he said, that's it, girl. The cameras left out the room, and it was just my dad and I. And I began to cry. And these was tears of joy because I felt in my heart that my journey was over with sickle cell disease, the pain, the suffering, all the what ifs, and that this was gonna be a new beginning for me. For my dad, he said, I don't know what it is, but God has something special for you. 2 weeks after, the side effects of the busulfan began to kick in. I lost my hair, which I was prepared for. Joke with my dad about how we were the same now because he's naturally bald. And but about 2 days after that, I got the sores in my mouth, which Dr. Frankel explained was a risk because of the busulfan will attack the soft tissue. And that was a doozy. I couldn't eat. I had one pill that couldn't be substituted for IV. So I had to take it, and it felt as though I was swallowing glass because everything was torn up from the chemo. I had a moment, it was like, what did I do to myself? But I had some of my most important people around me that assured me, Vicky, you seem worse. We've had times with we was knocking on death doors, so this is not gonna last always. So I held my head back up, and that didn't last for about 2 weeks, and that was over. My mouth began to heal. I experienced weakness, but I couldn't tell the difference between this weakness and the weakness that I already had with my sickle cell disease. That weakness had me bound to the house, but with this, I didn't need assistance getting in the bathtub. I was able to get out of the bathtub at the hospital, walk the halls, and I got discharged after the 30 days, and I stayed an extra month and a half in Nashville just so I could be close to the center. I was less than 5 minutes away from the hospital just in case because my immune system was compromised. But everything went without any hiccups. I ended up coming home to Mississippi earlier than expected, 2 weeks earlier, exactly. I was able to surprise my children. And that was a huge moment for me and for them to see my son. As soon as he saw me in my car, he just took off running in the embrace from my children with they were crying just to have me back home, and like my mom is here, and they knew I was safe, and I was able to walk back with them a little bit stronger. I didn't notice until a few months that things were changing because about 3 months, I was like, "Oh, I hadn't had a blood transfusion." And I was getting blood transfusions through apheresis every 3 to 4 weeks. I couldn't go no more than 6 weeks without having to have one, and this had been 3 months, so that's a milestone. And my kids, they invited me to the backyard to run with them, and I was able to run longer than I would have. I failed though, but that's because of my clumsiness, not because of my disease. But I still had aches in my body because I decided to do the taper. I told Dr. Rangel, I said, hey, how do I know I'm better if I still have on a fentanyl patch, I take Dilaudid and oxycodone? I said, I want to come off of this so I can really see. So we started the taper, and beginning of 2020, COVID happened, and then the shutdown, so I couldn't get the medications for the taper to taper properly. So I went through withdrawal, and that was ugly. It was body aches, chills, sweats. I was easily frustrated. I was at home alone with 3 small kids. They were like 9, 10, 11. No, yeah, 9, 10, 11. and trying to navigate school, virtual. I felt like the world was falling apart. But one morning I woke up and I felt absolutely nothing. I sat on the side of the bed. The room looked real bright. So the first thing came to my mind, am I dead? Seriously, I began to pinch my face. I began to pinch my thighs, and I said, "Oh, I can feel that." But still wasn't sure. So I called my children out of their rooms. I said, "Hey, y'all, come here." And they came out and was like, "What's wrong?" I was like, "Oh, nothing. I just want to see you." But I wanted to make sure they could see me, and I wasn't some spirit that was with unfinished business hanging on. So after they left out, that's when I realized, Vicky, this is it. It's really over. And I just praised God and I began to cry and I was thankful because this was the first morning I woke up that I wasn't tired after sleeping for 7 hours, 7 to 8 hours, and still tired. I didn't have to gasp for air. My body wasn't aching. I felt absolutely nothing. I felt normal. Um, and that continued throughout my life. So now it's been 4.5 years, a little over a half. July 2nd would make 5 years for me, pain-free. And I work full time. I'm able to travel and advocate, and I'm able to travel to see my husband during his deployments. I'm able to be a full-time mom, attend football games, be beside my daughter when she's cheering and doing majorette. I'm living now. I felt like I was just existing before, but now I'm living and enjoying life and I have a purpose. At first I gave up on dreaming because every time I dreamed, it got crushed and stopped, and my life had to pause continuously because those crises would hit me out of the blue. I couldn't time it, and my doctors was getting frustrated. Um, I was frustrated, but this is giving me better quality of life, totally different life than I ever expected. And I'm just grateful to God and to everyone who had a part in the science, manufacturing, everything, because this was life changing for me. Thank you all.
Thank you, Victoria. I mean, I'm so happy we have a documentarian in the midst here because that story you just delivered, the way you delivered it, the I'm honestly in awe of you. I'm honored to be in your presence and so happy you're able to participate in today's session. Thank you so much for sharing that story. It was beautiful from beginning to end. And I'm really happy to hear 4.5 years going on 5 years. You've been able to, you know, walk into your deserved life like you definitely talked about. So thank you so much.
Thank you for having
Gabriel,
me.
no, it's our pleasure. Over to you, my friend. I know you're going to talk us through a bit about your experience with bone marrow transplant.
Yeah, and thank you for having me, and shout out to all the speakers before me. And Victoria's story was amazing, got emotional a couple times. But yeah, I wanted to, I'll give you a rundown with my experience with the bone marrow transplant, which is a bit different from gene therapy like Victoria did. So just like as a background that like got me into the trial, um, I had a pretty big crisis in 2018, and that really like had a big impact on my body and my health. And like generally I've been like the type of person I've been like I would have my crisis a couple times a year or whatever, but I can bounce back kind of quickly in a couple weeks or so or whatever. But after that crisis, I felt like a dramatic shift within how my body was able to recover. It took me like almost over half a year to kind of like really recover from that incident. And to be honest, like I didn't feel like I fully recovered to how my body was functioning before. Like I had a new normal. that I had to get used to. But with that crisis, my doctor at TGH, my hematologist at TGH, asked me if I'd be into doing a bone marrow transplant. So I had known about the bone marrow transplants like as a curative treatment for sickle cell like way back because I did a deep dive like in my teenage years and I found out that they first did a bone marrow transplant to cure sickle cell like back in the 80s or something like that. So but after watching the movie 7 Pounds with Will Smith, and there's a scene where he donates his bone marrow to somebody, and they had to drill in his bone, and then he couldn't walk for like a while and whatever. And I did more research on like how bone marrow transplants works, and whoever the donor is, like they're pretty much like out for a year because they have to drill in their back or their leg or something like that. And it's a really long recovery process for them. Um, so I just never considered it because I wouldn't want any of my siblings to go through that. Like, I'm the one who has sickle cell, so it's kind of like my cross to bear. I don't want to have to share that with somebody else. Um, but with a bone marrow transplant, like you need a donor, like and most preferably like a related donor. And fortunately for me, my dad um had 6 kids. So there's a few people to test to see if they're a match, but actually only one of my sisters was 100% match out of everybody. And so that was kind of that was like a blessing. And I was kind of nervous to ask her if she would do the test to like, hear her response. But when I was speaking to my hematologist about like not wanting my siblings to do a test and have to go through that kind of bone marrow transplant process for the donors, he like explained to me that like they've come a long way with bone marrow transplants because they've been doing it so much with cancer patients that pretty much they don't do any drilling in the bone. They pretty much give the donor an injection that like helps them create more of the stem cells into and it gets released into the bloodstream, and then they pretty much just do like a uh they pretty much, I can't remember what the name is, but they essentially like take out the blood from them, separate the stem cells from the real blood, and then put it back in them. So it's kind of like a blood transfusion, and that's it. And it only lasts like they only have to do the injections for like 5 days, and after the exchange is done, like the recovery is like a day or so. So I was like, okay, cool. Like I feel much better about that. So told my sister, like she said yes, like without even thinking about it. And my sister that was a match, she's 2 years younger than me. So out of all my siblings, like we're the closest in age. So she's really seen my journey like throughout my life with sickle cell. Um, and I kind of want to give you like a more insight on like the process of like what happens when you're actually going to when you're trying to do these kind of curative treatments. So the you'll meet with the bone marrow transplant team or the gene therapy transplant team, and they'll give you a rundown and they'll be like, hey, this is a process. You have to do all the testing to make sure your body's right. Because also like your body has to be strong enough to withstand the treatment, even though and it's kind of weird because you kind of have to be like sick enough to qualify for the treatment as a trial, but be also strong enough to withstand the treatment. And I guess I just had happened to fit like in between that. And so they'll do a ton of appointments, like they're literally testing everything. So like the amount of appointments you have is like you're in the hospital like a couple times a month, maybe for like a year or like 6 months to a year in doing all these tests and stuff like that. So yeah, definitely expect like, time off work and and those kind of things in doing tests. And it's kind of exhausting, but like, there's a bigger goal that, you're trying to achieve. So, it's easy to get through. And also like, living with sickle cell, we're used to the hospital. Hospital's kind of like the second home, so um then they'll tell you about having to do some form of chemo and radiation. And pretty much they'll be using the radiation the chemo to suppress your own immune system. And then the chemo to suppress your immune system. Correct me if I'm wrong, Dr. Wall. And then the radiation to like um
Make
clear
space.
out.
Yeah,
Yeah,
yeah.
yeah, to make space for to empty out your bone marrow so new bone marrow can grow once you have the donor cells. So when you go through that process, it sounds pretty easy. Like you're only doing like chemo for like 5 days. There's a wet rest day in between. Then you do radiation, and then a few days later, it's your infusion day where they infuse your donor stem cells. And it sounds pretty easy and chill and all that, but it's actually going through chemo and radiation is a lot more hectic than I anticipated. But having sickle cell and dealing with the pain, it was like easy. I can like kind of understand like dealing with kind of complications and stuff like that. But it's a bit different with like the symptoms that you get. And for me, I had the chemo, and the night before the radiation, I had a crisis. So I guess my body was getting um stressed from like, the chemo that was happening. And also I was mentally stressed because the the day I got into the hospital to start the treatment, I found out that my dad was like at like stage 4 cancer. So it's kind of like out of the blue. So like I was like really stressed, but anyways, fast forward, I had a crisis the night before. But thankfully I was already in the hospital when the crisis was happening. So they were able to like give me pain meds and kind of like treat it right away where it was like at a manageable place. But they didn't want to move the radiation. Um, I can't remember the reasons, but it wasn't really a thing that we can like just move because we already started the process. Kind of like once you start going, you're going. So I did the radiation, and um it was very painful after. If like your whole body you feel like you're getting shocked to your whole body, and your body, like I had like swelling in my face, and my like you can tell that like something was going on or whatever. But it didn't last too long. It was kind of chill. Like after a day or two, it was fine. Um, and then when you get to your infusion date, it's honestly like pretty like anticlimactic because it's pretty much a blood transfusion, but you're just getting stem cells instead. But uh, when you do get when you do have going into your transfusion date, make sure you have some type of strong candy that you can eat because the the something that's in the preservative that's in the freezing mechanism that freezes the your donor stem cells makes you taste like a strong corn or something or whatever, and it tastes nasty. So have some sort of strong candy that you can have in your mouth or something like that to like kind of like offset it. And expect to throw up and whatever. But it's fine. You'll make it through it. But at that time, like in getting like the infusions, or like as a transplant is happening, like, you know, it was kind of like an unbelievable moment because it's like, you know, I've honestly been kind of skeptical about the whole thing until that point. Like I just didn't really take in what was happening until that point. And like um yeah, just like all the stresses and stuff and like what I've been through my whole life with sickle cell and all the battles I've had. Like, you know, I just got like really emotional. I was crying and my sister was with me. And I did this during COVID, like, or in the like 2021. So there was a lot of restrictions about like how many people can be there. So I had my sister with me. So yeah, I just like, you know, was crying in her arms. Just you know, getting emotional now, like thinking about it. But yeah, it was like a really amazing moment and stuff. So fast forward, I do the transplant. You're in the hospital for like about like 4 weeks after that. And they're monitoring your body and you're waiting for um I can't remember because it was a while ago now, but you're pretty much waiting for all your counts in your system to hit zero, and then for your body to reset itself at zero, and then it starts to go back up where the new cells are starting to grow and graft and all that stuff. So I pretty much breezed through that whole process. But something that I didn't mention earlier is that um, one of my brothers now started the transplant process a few weeks after me. So I was actually in the hospital with someone that was doing it at the same time, like we overlapped with each other. So as I was breezing through it, he wasn't breezing through it as well as I was. And even like something that Victoria mentioned, like with having like the mouth sores, he was getting the mouth sores and like a lot of it, and he would be telling me like, yo, like I can't eat, like my mouth like hurts and stuff like that. And I was like, damn, like, you know, it's I wasn't experiencing all the things that he was experiencing fully. Like you get really fatigued and I had a lot of headaches and stuff like that. And I would feel like some pains and like honestly like extreme fatigue. I was sleeping like probably like 70% of the day. But like with things like mouth sores and other like kind of like symptoms, I wasn't really feeling as well as much. But when the I later in the process I started to get those symptoms like the mouth sores. Actually, I think I got the mouth sores after I exited the hospital. So pretty much I breezed through the whole thing. I I was discharged from the hospital after being in there for like 6 weeks. And I was at home feeling good. I'm going on walks and you know, doing all the things, you know, and it's the pandemic, so you know, I have my mask on and being safe with where I'm going and stuff like that. But a few weeks after, like I was starting to feel like all the symptoms, the mouth sores came, I was getting infections, any germ was like making me sick. Um, because again, like you don't have an immune system really during this time. But fortunately, I a comment on the mouth sore things. So I expressed to the nurse practitioner, I'm like when I start to get the mouth sores, I'm like, hey, I don't want it to get to how my brother's mouth sores was. So like, y'all got to figure this out. Unfortunately, they found a solution that worked for me and him as well with some sort of like mouth rinse, which pretty much got rid of the mouth sores pretty easy. But um, from the time of like you know, six, like 2 months after the transplant, for like the another like 12 months, I was like having a lot of symptoms where I was having to go to the emergency because I was getting sick. I was getting infections like in my port and and all those things. And honestly, it was getting to a point where like I didn't really feel like this was working or you know like and there is a huge like risk with um when you're doing a bone marrow transplant with graft versus host disease developing and stuff like that. So it was taking so long for me to like start to feel strong. And my brothers who had faced a lot of symptoms early on were starting to like all their stuff was going up, like their hemoglobin was going up to like 140. Mine was still like 90s, and I was still getting blood. I was getting blood transfusions because my hemoglobin was dropping to 70. So and mind you, there was a few of us that have done the trial at Princess Margaret at UHN, and we all know have been connected with each other, so we all talk about our experience, and all of us have had a different experience with the symptoms we feel and when we feel them. So yeah, everybody was, breezing through it after, and like, their hemoglobin's strong and they feel strong and they're doing all sorts of activities and stuff like that, and I was still like feeling sick. But after like in spring of last year was when I started to feel a difference in my body and I started to feel strong. And I've been like, gravy ever since. And still on the medication and all that kind of stuff. But I think another big thing that with this process, like if you're considering this, is that there's a huge mental kind of gymnastics that you're going to be faced with. And they always say, like, to people with sickle cell, like, oh, don't let people like sickle cell define who you are and this and that. But like honestly, like sickle cell does define who you are. Like I'm sorry to say that, but it does, because every decision that we've made throughout our life has been informed with sickle cell in mind. So um, as you're going through this process and you're thinking, I'd even tell you about my mind state before that to backtrack. So before I never like thought long term because I just thought that there's a good chance that I would die like in my 30s or 40s from sickle cell. So my whole life, I've never planned long term. My whole objective was just to enjoy as much time as I have with the people I love on this planet. And anything that got in the way of that, I didn't care. Like I just wanted to and focus on having, a good time while I'm here. So when now you're faced with, a curative treatment, pretty much or at least for me, I haven't been thinking, like I've never made a serious 5-year plan or even 2-year plan or anything like that. Now all of a sudden I feel like there's like a whole bunch of life that I have left to live. It's kind of like when you have to do an assignment in school and you have to do a reading of an article and you think it's a short article, and then you read through the first like couple pages and then you're like, wait, how long is this? And there's like 20 other pages and you're like, oh my goodness. So that's kind of like how I feel, but like essentially you kind of go through an identity crisis because um everything's been informed by sickle cell all your life, and now suddenly over a matter of months that disappears, and now the decisions you're making are not really informed by sickle cell, and you have to find a new way to kind of like figure out what's motivating you and your reasons for doing things and whatever. And I'm still struggling with that, like trying to like, navigate that, but it's definitely a big hurdle to overcome. And I recommend, even though I haven't done this, but I recommend, if you are planning on doing this, doing therapy before you start and continuing through that process. Because yeah, it's a lot um, to forgo, just like mentally speaking. Um, so what else is there?
And don't feel like
Yeah,
you need to. Yeah, go ahead. Yeah, sorry. Go ahead.
no, what were you gonna say?
Oh no, I meant if you feel like you've reached the climax, don't feel like you need to extend. But I didn't want to interrupt, so go ahead, Gabriel. Yeah.
Um, so the other things too is that when you're going through this process, um like they might tell you like, all right, you'll be in the hospital for like, maybe a month or two. And then they might say you might feel fine within like, a year or so, or maybe less than that, 6 months, but it's depending. But really, they tell you that there's a 2 year kind of like period where they're assessing the graft versus host situation, if that's going to arise or not, and you're going to be on immunosuppressants that entire time. But in terms of like taking time off and planning this kind of thing and your finances, um, I would say to plan to be away from work for minimum a year and a half. And I say a minimum a year and a half is because um, as we know with sickle cell, stress is a trigger for your health, and we know that because of our triggers and stuff like that. So you don't want to put yourself in a position, if you can avoid it, where you're stressing about having to go to work and doing all those things and also be in recovery, and that will put extra stress on your body. So I say, like, give yourself at least minimum a year and a half to be fully away from work and stuff, so you can just focus on recovery. And especially because you don't know fully like the hurdles you're going to have to jump in that time. So really like try and set yourself up to be okay, to last that long. In Canada, like you can apply for disabilities and stuff, which kind of helps and stuff like that, but like it's really not enough at all. So yeah, take with that what you will, but yeah, please plan financially to be off work for a while. Um, and to give yourself put yourself in the best position to heal well. And also, eating very good goes a long way. Um, like eating healthy, eating a lot of greens and all that stuff goes a super long way in doing this kind of thing. Yeah, so also too, I want to, just extend to everybody that's listening, like, if you do have more questions that we might not have the time to get through during this session, like feel free to reach out to Skago and just ask for my contact, and I'm free for anyone, and give them a rundown on what's going on and stuff like that. But yeah, all in all, I'm good. I feel like I'm still not fully in the clear yet, like I'm still being seen, but I feel really good. Oh, and one thing I want to mention too
Mhm.
is that you might still feel. So my sister that was my donor was AS, so she did have the trait. But you might still feel like you might have like tiny crisis, but they won't feel nearly as severe as what you experienced beforehand. And it sometimes you will kind of feel like your body wants to start like trying to fight you and have a crisis, but it won't happen. It's like there's a blocker on it. It's kind of like when you're starting a car that has a dead battery and the engine's trying to go, and whatever, the new stem cells and the new cells you have is just blocking that from that sickle cell um car to start. So yeah, you might feel that time to time, and I do feel like, some things when I get sick, but it's not even close to what I was experiencing before. Yeah. Yeah,
Man,
that was all from.
Yeah.
Gabriel, every time you tell your story, it gets a new angle you present it to me, and I really appreciate hearing more and more. And I think the resounding fact from both your story and Victoria's is this mental fortitude you both displayed throughout your entire journeys, this unwavering commitment to living your best lives and seeing yourself through through sickle cell, like you mentioned, in spite of it, where you're living with it in spite of it, but at the end of the day, you're able to find these curative therapies that helped you get to this new horizon. I'm just so happy to hear from you both and really appreciate you sharing your time with us. So we have about 15 minutes left. I wanted to there's a couple questions, a couple hands raised. I want to at least try and ensure folks had a chance to interact with you. The first question that came through was I was in respect to gene therapy, and I'm hoping maybe Dr. Wall, you might be the best person to provide a response. It says, my question is related to gene therapy. This person's sister had a buccal swab and was taken as a potential donor in 2022. To this day, we haven't received any feedback on whether or not there's a compatibility there. They want to know how long it takes to know if a donor is a potential match and what strategy or is there what strategies can a patient use to essentially get that point of clarification?
Yeah, so the testing now, it used to take 2-3 months to get that testing back, but now it's coming back in a couple of weeks. So for sure, the results are there. So you need to follow back with your hematologist who did the testing, and then sorry, just to re-engage. We have just over the last 5 years, there's been a real change in interest in transplant with the sickle cell community. So we have a whole bunch of patients who had started that process of testing and then kind of backed away, and it just wasn't the right time. And so I would recommend getting back in touch with your hematologist and getting the results and then referring into a program to get more information.
Beautiful. Thank you, Donna. There's another question here from an individual with respect to, so there's two questions, one with respect to BMT and gene therapy. And I'm wondering if we can, in response to this question, really distinguish the difference between the two. Because this person's asking, they're in Toronto, and they want to know how they can benefit from CRISPR in Canada. So I'm not sure if Dr. Wall, you can maybe start with that, and maybe Victoria, Gabriel, you can help folks distinguish between the two treatments.
Yeah, and then my colleague, Dr. Matthews, also is part of this journey.
Mm-hmm.
Because the trials for the first trial that was launched has closed to new patients because they've treated enough patients and are getting the information going forward. There is another trial that is opening in Canada at several sites. And so I would talk to your hematologist, and they'll be able to to find a site. Right now we do not have a study open at Sick Kids, but however, the CRISPR Vertex folk and the Bluebird folk are, I'm sure, going to be looking at making their proposal to Health Canada. I believe the CRISPR the Vertex one is going in the next month or so for is there enough data to support being approved in Canada, and then that'll be followed by an application to CADIT if it's not already happened. So that whole journey, so we're making the switch from doing something on a clinical trial that's sponsored by the company to this is now a treatment that should be offered in Canada. And it's a very complicated answer to that. I hope I was able to tell you in my story. There's a lot of things at play. Does that help?
Definitely. Matthew, anything you want to add to that?
I'm just trying to find my mute button. No, I think that covers a lot of things.
Perfect. Okay. And Victoria, did you want to try and give folks a quick distinction between the two?
I noticed a few differences during Gabrielle's talk. With me, I didn't need a donor. They used my own cells. And another difference, I didn't do radiation. I only did chemo, and it was a strong dose of chemo to clear the pathway, so I didn't have to do both. And I didn't experience any vomiting or weakness that was more severe than my sickle cell pain. I didn't, my sickle cell weakness. So vomiting, I experienced a lot of nausea, but Dr. Frangoul, they treated me for the nausea to prevent the vomiting. And one detail, when I wasn't able to eat for 2 weeks, I did receive my nutrition through IV. So that kept me from being weak, from not going without food and water. So there's a few differences there for me that I noticed between bone marrow transplant and gene therapy. And then the graft versus host probability doesn't exist in gene therapy because they're your own cells. And it was one other thing I wanted to mention, but again, flew out the window, my memory. But I want to tag team with Gabriel about the mental health. So I did prepare sickle cell warriors. We have to be honest with ourselves and know where we stand mentally, because it does affect us. So I did seek therapy before the going into this, but it wasn't before gene therapy. It was when I initially thought about doing a bone marrow transplant. So I knew I needed to go in mentally strong. And the hospital provided me with a therapist during the procedure. So I had a therapist while I was in the hospital going through gene therapy treatment. It stopped once I got home and when the COVID shut everything down, pandemic. But I did pick it back up myself because I knew it was something I needed. You do go through a identity crisis after you recover, because what do I do now when you have this pressure? What do people expect me to be doing? I'm behind. I didn't get to finish my dreams. I didn't get to complete college. I completed, but not in the field that I wanted to complete it in. So where do I start? So what's next? It's a lot of downtime at home and you're looking at the wall, still feeling trapped because I wasn't able to work before. So I had to do something completely outside of my normal, even though I was fulfilling my dreams. It just happened so fast to where it was a shock. So don't be afraid to reach out and get the help you need to, because we come too far to let our mindset be the thing that calls us to sink. So it's nothing to be ashamed about. Having someone to talk to about what you're dealing with, and finding the right therapist that can truly understand that there's a journey. And I call it PTSD after having sickle cell, the trauma, because one thing that used to trigger my crisis was changing weather. So every time I live in Mississippi, so the climate change quick and you don't know what to expect. We can go from hot to cold real fast. And so when that started happening, I was like, oh, we under tornado watch, or it's about to freeze, and am I gonna have a crisis? And I would start feeling sick, not the pain, but sick to my stomach, just worrying about the what ifs. And oh, you're the first person, so it's not guaranteed how long this is gonna last. But that's when my faith come in along with my therapist to reel me back into what's my new reality now, that I don't have to have this hovering over my head. So it's important to seek the treatment that you need.
Yeah.
Perfect.
And to
Go
piggyback
ahead.
off Victoria, there is like with that PTSD, and there's honestly like a kind of like a paranoia that, we as sickle cell patients have in like, we don't know when a sickle cell crisis is going to happen. So we're always trying to anticipate and trying to do things that will kind of prevent us from bringing on a crisis. And like now that I don't have a crisis like that, I'm starting to notice things that I do, just mannerisms that I have, because I was scared of getting a sickle cell crisis. For instance, when I got out the shower, instead of wrapping, as a man, wrapping a towel around the waist, I would wrap it around my chest because I didn't want my chest to get cold, and get a chest crisis. So I would always do that. So what now after having this, I noticed, oh, like I was doing that because my chest was I was scared of my chest being cold and the body temperature changing too fast and getting a crisis. Even as far as, and my girlfriend kind of pointed this out, is that I move my toes around a lot. And I noticed, like, like if someone were to punch you or something like that, or hit your arm, you might clench your fist or your hands. And I used to get a lot of pain crisis in my legs. So I'd always be clenching my toes. So that's something I still do a lot. Like I'm always like clenching my toes and stuff, like, and like moving them around, as like it's like a pattern that I've like developed over the years. So like you'll start to notice like these kind of things that we've dealt with mentally, and we've have a bunch of coping mechanisms that you just don't need, going further. So yeah, it's a process to get over it.
Perfect.
Can
Thanks,
can
Gabriel.
I reply to Roxanne? It's a message that I saw earlier that I can't find now, but she
Um,
said that she
I've
was
seen that.
happy
Yeah.
that we was able to do better, and for some people, they don't qualify. A part of my story was when other treatments came along, I signed up for other studies, and it was always I didn't qualify, I didn't qualify. So don't give up hope and feel like you'll never qualify for anything,
Hmm.
because I qualified for this and this was my path. So something for you will come along, and things are changing, it's evolving, medicine is changing. There's so many advances. So one day you're going to qualify for something. So don't give up and feel like there's nothing out there for you. No help is going to come, because I was in that place. So I can truly understand. I was like, every time I sign up, I don't qualify because I just had a blood transfusion. I don't qualify because I've been in the hospital too many times. I'm not going to get anything. But I qualified for this. I was sick enough and yet healthy enough at the same time. So something is going to come for those who may not qualify for CRISPR, or those who may not qualify for BMT. There's going to be something in the making, in the works. We have a voice now. That's what I like to say.
Wow. Power to the people. I love it. So there's a poll, folks, that's currently active as well. If you haven't been able to submit your responses, please do. That will be live until the end of the session. Lisa's been having her hand up for quite some time in the depths. I want to give her an opportunity to pose her question. Oh, never mind. Lisa put her hand down, I see. Never mind. So back to the questions I see in the chat. One person says here, just going back up. So it's just from Diane. How long will patients be checked for leukemia after treatment? Will this be an ongoing thing, or when are these side effects? Well, when will the side effects stop being assessed?
The quick answer is never. And so because you're going to be the reporting person, it's you, and you're going to know if you have a pain crisis or not. And so with Health Canada, with the first trials, we were asked to do 15 years of reporting just because it's a brand new treatment. And it's a annual reporting in. Do you have any pain crises? Do you have, and check blood counts on it. As part of the study, there's been several bone marrow checks that are done, and be very careful looking for any trouble. And so far, there's been no trouble, but we're still very early, and there's small numbers of patients treated so far. So it's going to be followed long-term as part of your regular care.
Perfect. And this might be the last question, unless folks can stay for just a minute or two, but I want to be conscious of your time. It is a Saturday, but this is for you, Dr. McDonald. After extensive review of a new medication, has there ever been a recall of said medication? And how long do you check for major changes or submissions for a new medication? Does it vary on the type of medication?
Sure. Yeah, thanks for that question. I think it would just, the answer is really kind of extends on what Dr. Wall was talking about in terms of safety of any drugs and monitoring it as a continual process. It never stops.
Mm-hmm.
Any side effects and that kind of stuff can be found online. Health Canada's product monograph provides a list of, adverse events, what side effects you could expect. There's also online reporting that's kind of always active. There's an active database where adverse reactions to drugs can be entered, and it's continually tracked by Health Canada. And then CADF has a number of different programs that also look at safety questions over a period of time, like our post-market drug evaluation team looks at real world evidence around safety of medications. That's a very common question they get. So I think a short answer is it's continuous, and I don't think it ever stops.
Fantastic. Okay, and there's one final question right now. I want to make sure we pose it before putting my email in the chat for folks who may have other additional questions. It's nice to see and hear that these curative therapies are being done on younger adults. Are there any studies being done on older adults? And if so, are there any cutoff ages for these particular clinical trials?
Yeah, so you got the pediatrician speaking. So the first trials went to age 35. There, I can for sure, with the development of the treatment, there will be questioning, and a trial is just a question, questioning whether we can safely treat older adults. And I think the real issue is going to be what kind of complications you've had, how severe, how well your heart, liver, kidneys are working. And so that's going to be what you're seeing today is our first experience. And so from here, we then extend older, younger, sicker, healthier, and trying to find really what is the right time to do the treatment. So we're early days.
Early days. But that's it. A promising future is ahead of us, folks. So do not lose sight. I think you should all take stock of what we heard from our lovely guest today, that resilience is obviously a staple amongst many of us here. And the longer you continue to be steadfast and push on forward, it would be fantastic for you to, reach these same horizons that many of us have been able to, be privileged to hear about today. So with that, I just want to give a big thank you to each of our speakers, especially you, our audience members. Thank you so much for taking 2 hours out of your Saturday afternoons to join us today. That draws to a close our Chicago Learning for Life session. Once again, the poll will close pretty momentarily. So if you want to submit any feedback, that's the best place to do so. I've entered our email, there's my email into the chat. So if you have any other questions that we weren't able to respond to this afternoon, please feel free to email me there. Thank you, Gabriel. Thank you, Dr. Wall. Thank you, Dr. McDonald. And thank you, Victoria, so much for joining us and sharing your insight and experiences. We cannot thank you enough. So with that, have a wonderful rest of the weekend, folks. And we will look forward to seeing you at the next session in May. Take care.